Hello everybody, I'm Chen. I'm going to briefly introduce four metrics for summary statistics and for genome-wide association meta-analysis. The details may be found in our publications entitled Across-cohort QC analysis of GWAS summary statistics from complex traits, published in European Journal of Human Genetics. Let's look at the experimental design of a GWAS first. In GWAS, there is a central hub. Each cohort conducts GWAS and sends the summary statistics to the central hub. So actually, the GWAS central hub only receives the summary statistics from the cohorts. The risk for GWAS is garbage in, garbage out. So how well a central hub can test the quality of summary statistics is crucial for the success of a GWAS. We propose four metrics and visualization tools for the central hub of a GWAS. Both FPC and meta-PCA can use demographic information to validate the allele frequency information. Lambda meta can detect the proportion of overlapping samples between cohorts. Pseudo profile score regression can pinpoint overlapping samples between cohorts. Let's validate the quality of genotypes for GWAS first. We know genotypes mirror geography. So intuitively, if we have reference cohorts from Europe, Asia, and Africa, then the received cohorts will have a relatively short distance to the reference cohort of the similar origin. For joint metabotrip cohorts, we measure each cohort's genetic distance to these three reference populations representing Europe, Asia, and Africa. We see three categories of FST patterns to the reference population. Then, for each cohort, we convert FST values to its coordinates in what we call the FPC space. Actually, we find each cohort eventually is located close to the reference population of the similar origin. It is expected and consistent to their demographic information, so we believe the quality of genotype is good. Principal component analysis is commonly used to examine the quality of genotype data for GWAS. We also apply the PCA for summary statistics, but compared with conventional PCA for individual level data, we replace the vector for individual level data with the received allele frequency for each cohort. We call it meta-PCA, which is a second metric introduced. Look at here. Left is the European map, and inside is a triangle that connects Sweden, Finland, and Italy. The panel at the right side is a projection of 174 joint height cohorts using meta-PCA. In the meta-PCA plot, we are also able to find a similar triangle connects these three countries. Using allele frequency only, meta-PCA can capture the topology of a European map at a pretty fine resolution. Let's examine more details in this plot. We can find that cohorts from Canada and France have very similar coordinates in meta-PCA plot, reflecting their similar genetic background. We checked the demographic information of the Canadian cohorts. Actually, they are from Quebec, and we believe its French heritage leads to this similarity. We also find cohorts from Australia and the UK have similar coordinates too. We believe it is true because of continuous gene flow from UK to Australia in the last several hundred years. For the Italian cohorts, we can see one cohort stays very close to down left corner and away from the reference Italian cohort, which is from Tuscany. The standing out cohort actually is from Sardinia Island. For large-scale GWAS, one of the concerns is sample overlap between cohorts. How can we infer that with summary statistics only? We use lambda meta to do the job. Lambda meta is constructed on a pair of cohorts over their aligned summary statistics, such as genetic effects and sampling variance. When there are no overlapping samples, lambda meta should follow a chi-square distribution of 1 degree of freedom. Once there are overlapping samples, its distribution changes. The technical details may be found in our paper. I only introduce the results here. The panels atop represent distribution of the lambda beta when there are no overlapping samples. We can see the slope of 1, the blue line. When there are 100 overlapping samples, 10% of the total samples between two cohorts, the slope turns down. Please compare the blue lines atop and at the bottom panels. We can see the difference. Let's look at the real data example. We downloaded the GWAS summary statistics for PGC Psychiatric Genomic Consortium. PGC Phase 1 has 21,000 samples, and PGC Phase 2 adds another 18,000 samples into Phase 1. Then it carries out lambda meta of 0.257, indicates substantial overlapping samples. We can see the red slope, which shows very clearly how significant the overlap is. Actually, PGC2 completely covers PGC1. If we don't know the overlapping samples for PGC1 and 2, we also can use the overlapping slope of 0.257 to estimate the overlapping samples between these two cohorts. We applied lambda meta to 174 joint height cohorts and get a matrix plot. Every point in the matrix indicates a lambda beta for a pair of cohorts. The upper and the lower triangles represents lambda beta greater or smaller than 1. The overall distribution of all these lambda beta is nearly a normal distribution, with a mean of 1.013, very close to 1 but slightly larger than 1. It may be due to various protocols adopted by cohorts in conducting GWAS. In particular, we pick up the largest and smallest lambda beta detected. The largest one is 1.247 and the smallest is 0.886. However, lambda beta only detects the proportion of overlapping samples. Can't really tell exactly which individuals are shared between two cohorts. Without breach privacy, pseudo-profile score regression can do the job. We call it PPSR. In PPSR, the regression coefficient is 1 for identical individuals, blue points, and 0.5 for the first-degree relatives, yellow points. Using 55 PPS, the left panel demonstrates the shared controls are detected between WTCC bipolar disorder and coronary artery disease. Then, in the right panel, with 265 PPS, it is even able to find two first-degree relatives. This study would not be possible without the support from many colleagues and grants. Thanks very much for your attention.